Thank you. I want to thank Kat and the organizers for this. The invitation is really nice symposium. So, I thought it was working. Okay. So, our lab is generally interested in how organs form in embryos and how they renew and regenerate in adults. And specifically, we've been, over the past few years, really interested in understanding how epithelial stem cells enable regeneration of adult epithelia. And we look at two main parts of the body, the craniofacial complex and the intestine. So I wanted to tell you two short stories today that we recently published, and the first one is about intestinal stem cells, and the second one is about oral mucosal progenitors. And the general theme is understanding how these different epithelia can respond to damage and how they can demonstrate plasticity and complexity under these various conditions. So the first story is about the intestine, and as I'm sure most of you are aware, the intestinal epithelium is one of the most regenerative tissues in the body. It turns over in both mouse and human every several days. And in the small intestine, we have the sort of business end of the epithelium, or the villi, which is where absorption and secretion happens. And the progenitors are at the base of these units called crypts. And this is a cartoon of a section through one of these crypt villus structures. And it's been known for a long time that there are progenitors that are present here, as I said, at the base of the crypt, that then give rise to cells that terminally differentiate and then migrate up the tip of the villus, and within a few days are shed through a process called anoikis. For a number of years, there was controversy about where exactly the stem cells resided, but to summarize a lot of work, I'll say that under homeostatic conditions, it's been clear for a number of years that these relatively rapidly proliferating cells called crypt-based columnar cells, or CBCs, which are present at the base of the crypt and sandwiched in between secretory Paneth cells, which are the cells in red, are the workhorse stem cells of the gut, and these express a gene called LGR5, which is part of the Wnt signaling pathway. For those of you who are aficionados, there's been a fair amount of controversy about another cell type called the +4 cell, or the putatively label-retaining cell. If there are questions, we can talk about that, but I'll just say our opinion on it is that at least under homeostasis, it's really this stem cell here, which is the main stem cell in the gut. So about 7 or 8 years ago, we began a collaboration with Fred de Sauvage's lab at Genentech, who had made this really nice allele which enabled ablation of the cells that express LGR5, these crypt-based columnar cells, by knocking a diphtheria toxin receptor into this locus together with a GFP reporter. And this image here just shows you in green the LGR5 expressing cells, and in red is a marker for proliferation. And so there are, I forgot to mention this, but there's a group of cells called transit amplifying cells, or committed progenitors, which are present above the base of the crypt, which are derivatives of the stem cells. And you'll notice that also the stem cells themselves are Ki67 positive, so they're also proliferating. And so the hypothesis here was that ablating the LGR5 expressing cells would have catastrophic effects on the gut because of this idea that this is really the workhorse stem cell in the intestine. So we were actually really surprised when we killed these cells, and over this is a period of 10 days, so a couple of rounds of renewal, this is histologically, but we did many other assays and found that there was really no effect to the homeostasis of the intestine by removing these cells. And yet, when we give the toxin for several days to ablate these cells and then withdraw it, we see that the LGR5 expressing cells come rapidly back. So this is after a few days, there are no LGR5 expressing cells, and then within about 3 days, they repopulate the base of the crypt. So the gut likes to have these stem cells there, but it can, for at least a few rounds of renewal, function perfectly fine without them. And it was these experiments, which we did a number of years ago, which got us interested in a lot of the questions that we're asking now, including what regulates this really remarkable plasticity, the ability of the gut to function in the absence of these stem cells. How do the stem cells, ISCs is the summary for intestinal stem cells, how do these respond to injury and inflammation? And then in the long run, what we'd like to do is understand if we can harness some of these regenerative powers and some of this plasticity in order to treat disease. So the story that I'm going to tell you now is one that we published a few months ago. And it has a little bit of an unusual entree, so I'll tell you how we got into it, which is to try to use an infection model to understand this plasticity. And the model that we use is helminths, which are a natural parasite that infect essentially all animals in the wild and many people in the developing world. And we as animals have co-evolved with these parasites, and the response to them is often not it's often non-symptomatic. They can cause malnourishment or anemia, but they're generally not very significant effects. And the infections, while they've been largely eradicated in developed countries, I'll show you in a minute, this has some unintended consequences, perhaps. As I mentioned, the immune responses to the helminths are the result of this long coevolution between the host and the parasite. And the response tends not to kill the parasites, but to repel or restrict them. So what might be these unintended consequences of elimination of parasites in the developed world? If you look at a map of where there's a high incidence of autoimmune disorders versus a high incidence of helminth infestations, you'll see that they're kind of opposite. And while this is obviously just a correlation, there is some evidence that there's some connection between these two issues. And this is part of what's known as the hygiene hypothesis, the idea that we grow up and actually prenatally develop in an overly sterile environment. And one example of how this might connect with the intestine is a story that was published a few years ago from Mike McCune's lab at UCSF, in which a patient, I think this guy was actually a postdoc at UCSF, who had inflammatory bowel disease, traveled to Southeast Asia and self-medicated with parasites. And so you can see here, all these little squiggles are parasites that are present that he self-administered, and the yellow goop is the inflammation. And this is in 2007, and then over time, the inflammation is resolving. So this is just one example, and our hope is that we can figure out less disgusting ways and more mechanistic ways to understand how this works, and perhaps harness some of this regenerative ability of the gut. So the model that we've been using is a natural mouse helminth, which infects mice out in the wild, and it's called Heligmosomoides polygyrus. And like many of these parasites, has a really unusual life cycle in which the larvae will, after infection, burrow in through the epithelium into the stroma, and then they'll live there for about a week, where they encyst and molt a couple of times, and a granuloma, which is a large collection of immune cells, will form around them. And then after about a week, the adult animal will emerge into the lumen and live wrapped around the villi for the rest of its life. And so this is a histological slide after one of these infections, and this is the granuloma. This actually here is the worm snaking in and out of the plane of the board. And this is a stain for immune cells, showing you how rich the granuloma is in these myeloid cells. So it was known for a long period of time that the crypts that are right around this granuloma are actually hyperproliferative, that you see more proliferating cells there than you would in the wild type, or in the uninfected animal. And so the hypothesis that we had at the beginning of this project was that there was something either made by the granuloma or by the worm that caused hyperactivation of the stem cells or the progenitors in the base of the crypt. And so this is a project that was done by a fantastic grad student, Brenes, who graduated recently, in very close collaboration with Adam Savage, who was a postdoc in Rich Loxley's lab and is now in Seattle. And so the first thing that Brenes and Adam did was to take these LGR5 reporter mice, the same ones that I showed you before with the GFP knocked in, and infect them with the Heligmosomoides larvae. And they got a really surprising result, which was the opposite of what we had expected. So rather than seeing a hyperactivation of the stem cell program, what they saw was that this marker gene, LGR5, is actually turned off in the crypts that are right above the granuloma. And I hope you guys can see this with the light. Okay. So whereas those crypts that were at a distance maintained LGR5 expression. So we thought, okay, well, maybe this is something specific to do with this LGR5 reporter allele. So we looked at this surrogate marker called OLFM4, which is a notch target that's expressed in these intestinal stem cells. And here again, we saw that those crypts above the granuloma have completely turned off OLFM4, whereas those crypts that are at a distance have kept it on. And so then we went back and confirmed what I had told you before, which is that, yes, indeed, although they've turned off the LGR5 and OLFM4, these two canonical markers of the stem cells, the crypts that are above the granuloma, and red here is EDU, which is a marker of proliferation, are actually hyperproliferative compared to those crypts that are at a distance, and that's quantified for you here. And so at this point, what Eisbrand decided to do is a fairly heroic experiment, which was that you can actually see each of these little granulomas here are these little red dots. So he went through and punched out hundreds of these granulomas and then sorted out the crypt-based epithelium and did an RNA-seq experiment. And he got a really nice data set. And there were a couple of really important things that came out of this data set. One of them was that, so these are the granuloma samples, and they're compared to the non-granuloma samples from the same mice. So one of the things that he saw was that it wasn't just LGR5 and OLFM4 that were downregulated in the granulomas, but actually these green genes here constitute the transcriptional signature of the stem cells. So the entire suite of genes that, or almost the entire suite of genes that are thought to mark these stem cells are downregulated. So it's really turning off a whole program of gene expression. He also saw that there were a number of genes that were upregulated in the granulomas, and we focus particularly on this gene Ly6A, which you heard about earlier today. This is another name for the gene SCA1, which is a marker of a number of different adult stem cells, but hadn't really been studied in the context of the gut before. So fortunately, there's a really nice antibody for SCA1, and Eisbrand and Adam then went in and stained for it, and they saw that indeed SCA1 is upregulated in the crypts above the granuloma compared to those that are at a distance. Which they also confirmed using flow cytometry, and actually see that SCA1 is upregulated very fast within a couple of days of infection, so it's one of the earliest markers of infection. And also, as I'll show you in a minute, it gives us a really nice handle to sort the cells. And I won't get into this for the sake of time, but we also found that other injuries will produce similar effects. And so this is not something which is restricted to just this worm infection, but is some kind of general response to damage. So as I said, having this SCA1 now enabled us to sort the cells. And so Eisbrand took these mice that were infected and sorted out with SCA1 positive were the crypts that are above the granuloma, and the SCA1 negative, he could sort out the LGR5 expressing cells. So if you take LGR5 expressing cells, the wild type intestinal stem cells, and put them in culture, then you can grow out little mini guts, which are organoids, similar to the brain organoids that we heard about earlier today. So Eisbrand then took both the granuloma-associated crypts and the crypts that are at a distance and put them in culture in Matrigel, and he got a really surprising result, which was that the SCA1 negative cells, essentially the normal LGR5 expressing cells, just as we had expected, formed these organoids. But the SCA1 positive cells that were associated with the granulomas formed these huge cystic spheroid structures. And so we were kind of scratching our heads wondering what these things are. And then we came across a couple of really interesting papers which showed that if you take fetal epithelium and put it in culture, then you get the same kinds of spheroids that we see, in contrast to the postnatal epithelium, which gives you the organoids. And actually, if you quantify this ability, you see that in white here is spheroids and in gray is organoids, that it's really a nice linear decrease, that the older the animals get, the less they have the ability to form these spheroids in culture. And so this then suggested to us that perhaps what's going on here is that we're triggering in some way a fetal gene expression program in response to the injury, to the infection. So then first, Eisbrand compared the gene expression using qPCR in the spheroids that we got from the infected mice with fetal-derived spheroids. And he got an almost perfect correlation. So all the genes that are up in the fetal cultures were also up in the spheroids derived from the infected mice, and all the genes that were down were also down in the spheroids from the infected mice. And just for the aficionados, I'll mention that you can get these kind of cystic structures if you put organoids in high Wnt media, but actually we don't see an increase in Wnt signaling present either in the in these infected spheroids. So then Eisbein went back to the data set that he generated from the in vivo punch biopsies that he had done and did gene set enrichment analysis comparison. And here what he saw was that just like what we saw with the organoids, that there was an upregulation in fetal genes, that the granuloma was enriched in fetal genes, and that it had a decrease in the adult genes. And so the final experiment that he did was a single-cell RNA-seq experiment in which he sorted about 20,000 SCA1 negative cells and about 7,000 SCA1 positive cells from a single mouse, which was infected with a parasite. And this was a really interesting initial experiment. Now we're doing a lot of work in the lab to follow up on these studies, which was that most of the clusters that he got from this single cell analysis had combinations of both SCA1 positive and SCA1 negative cells in them. But there was this one cluster here, which is the cluster 12, which was composed almost entirely of the SCA1 positive cells. And it turns out that this cluster is the one that's enriched in the fetal gene. So this is the one that we're now going after for future studies to try to understand the specific cells that undergo this conversion to the fetal gene expression program. And so to summarize this first part, what we see is that the worm induces this hyperproliferative response in the granuloma-associated crypts, and this represses the normal gene expression program that we see in the stem cells. I didn't mention this, but we see that one of the drivers of this is actually interferon signaling, which is what promotes SCA1 expression. As I mentioned briefly, there are many other injuries that have overlapping and similar responses, and so now one of the questions that we're asking is, how are these responses similar and different? And most importantly, I think, is this idea that this fetal program is activated and is driven by this relatively small subset of cells within the larger SCA1 positive pool. And so the model that we have proposed is that in addition to the normal homeostatic program in which the LGR5 expressing cells make progeny, there's also this idea, which I didn't talk about a lot, but that you can either activate potential reserve stem cells, and we know that actually cells can de-differentiate under injury conditions. But now what we see is that there's yet another way that the crypt can renew, which is what we're calling developmental reprogramming, in which there's a wholesale shift in the gene expression program from what you see under adult homeostasis to one that looks like the embryo. And this program persists for as long as the injury is in place, and after the injury is resolved, then the program shifts back to the adult state. And so for the last few minutes, I'm going to change gears and tell you about another really interesting epithelia that we've been working on. This is work that was done by Kyle, another recently graduated student from the lab, who's a DDS PhD student, so he was very interested in the oral mucosa, which I didn't know very much about until he started telling me about what an amazing tissue it is. And if you think about it, it's sort of like the skin on the inside of your mouth, except for it doesn't have the adnexal structures like the hair follicles and the sweat glands. But if you've ever bitten the inside of your mouth, you know how quickly the oral mucosa repairs compared to your skin. So it has this really remarkable healing ability. And it's actually used quite a lot in tissue engineering and autologous transplant applications. And there's many diseases and cancers that are important. And I just put up this cartoon from a review from a few years ago to remind me to tell you that there are many different regions in the oral mucosa, and each of them have specific biology associated with them. And so this is a section through the oral mucosa. And the basal layer is the proliferative layer, and then the cells from here as they become superbasal, will differentiate and then finally produce a cornified layer. So one of the first questions we wanted to ask were very simply to describe the system. What are the dynamics of renewal in this tissue? And we had been focusing on a gene called BMI1, which is expressed in a number of different adult stem cells in other tissues. And so we decided to look where BMI1 was expressed in this tissue. And we saw that this is using a technique called RNA scope, which is a single molecule mRNA in situ. And so all these little red dots that you see here are individual mRNA molecules. And what we saw was that BMI1 is expressed in all regions of the oral mucosa and basically labels all of the oral epithelial, the basal oral epithelial cells. And then we used a BMI1 Cre-ER to do lineage tracing, which we got from Mario Capecchi's lab, crossed with RFP reporter. And this is showing you after a single induction of Cre activity, a number of different time points up to more than a year, essentially the lifetime of the animal. And so, as you would expect, if you're labeling the basal layer, if we label these cells a single time for the lifetime of the animal, they will then produce progeny. And we also, in work that I won't show you here, I should mention this was published very recently, we saw that a number of other genes, including keratin 14, which is a basal cell marker, and several other canonical stem cell markers are also expressed in the oral epithelial basal layer and label progenitors in a similar way. And so then we wanted to figure out how rapidly does the epithelium turn over and what are the cycling kinetics of these progenitor cells. So first, Kyle did a BRDU Pulsechase experiment, and he saw that the cells turn over relatively rapidly. So within the cell cycle is about a couple of hours. It's one of the fastest ones that we see in the mouse, and then they move up. So within less than a month, all of the tissue has turned over. And another way to look at this is using the keratin 5 H2B GFP label retention system devised by Doina Tumbar and Elaine Fuchs, in which we label all of the basal layer with GFP, and then we watch to see how long does it take to dilute out the label. And what we see is that within about 3 days, the label is getting diluted, and within about a week, there are almost no label-retaining cells present. So that's similar to what I showed you on the previous slide, evidence that the cells are turning over relatively rapidly. So then we wanted to ask whether these cells follow what's called the invariant asymmetry or the population asymmetry models, which these terms might not be familiar to all of you, so I'm going to show you on the next slide cartoons to explain what they mean. The invariant asymmetry model is the classical stem cell model, what we all think about when we think of, for example, a hematopoietic stem cell, in which every time a stem cell will divide, it will give rise to another stem cell as well as a daughter that is going to commit to differentiate. However, it's been shown in a number of different tissues, such as in the intestine and the esophagus, that there's another way that stem cells can function, which is called the population asymmetry or neutral drift model. And in this model, the asymmetry that is reflected in this cartoon doesn't have to happen every time a cell division occurs, but rather it happens at a population level. So the asymmetry exists in that you keep the progenitor population replenished, but each time a stem cell divides, it can give rise to 2 daughter stem cells, 2 daughter committed progenitors, or undergo one of these classical asymmetric divisions. And actually, you can model this pretty nicely if you do what's called quantitative clonal analysis. So there are predictions you can make based on these 2 different models, that if you were to have population asymmetry, the number of basal cells per clone should increase over time, because you can have stem cells that give rise to 2 more stem cells. Whereas in the invariant asymmetry model, whenever a stem cell divides, it only gives rise to 1 other stem cell. So the number of basal cells in each clone should stay the same. So when Kyle modeled this in the oral mucosa, he saw that this perfectly fit this population asymmetry model. And similarly, another prediction that we would make is that the number of clones over time should decrease in the population asymmetry model, because you can have some stem cells that will give rise to two differentiating daughter cells, and that clone would therefore be lost. Whereas in the invariant asymmetry model, since each time a stem cell divides, it always gives rise to another stem cell, the number of clones should stay constant. And here again, Kyle got a perfect reflection of the population asymmetry model. So these data really conclusively show that the oral mucosa, like the esophagus and the intestine, undergoes this neutral drift kinetics. Now, Kyle was also able to model the kinetics under a couple of perturbation conditions, and he saw that when he looked at old versus young mice, the old mice also underwent this neutral drift kinetics, but the clone sizes increased more slowly, which may have some connection to why, as animals and humans age, their ability to heal is decreased. And this is something that we're going on to study now. And another interesting finding that he had was that when he used 5-fluorouracil, which is a chemotherapeutic that kills proliferating cells, he saw that actually the clone size here in the basal layer increased after giving 5-FU. And we're still trying to figure out why this is, but the reason we think is that when you clear out the basal layer, you have more horizontal divisions, which then repopulate the basal layer. And so just to wrap up here, the last experiment that Kyle did was a single-cell RNA-seq experiment. And what he saw was, this was in collaboration with Ophir Klein's lab at Harvard. So he sorted the basal layer and then did single-cell RNA-seq. And what he saw was that within the basal layer, there were actually there were a number of clusters that expressed the typical basal layer genes, but there were also some that expressed genes that were more typical of suprabasal layers. And he then went in and looked at this with two-color single molecule RNA scope in situs and saw that indeed he could find many cells that were in the basal layer, for example, this one here, which were beginning to express very high levels of suprabasal as opposed to basal markers. And so the model that, and I'll summarize here, that Kyle came up with was that what's going on in the basal layer is actually that there's a lot of differentiation which is happening before the cells lift off and become suprabasal. And so we see that the oral epithelium shows one of the fastest turnover rates, as I showed you with the BRDU and H2B GFP experiments. It follows this population asymmetry model. And the single-cell RNA-seq demonstrated that there are progenitors that are at various stages of maturation that are present within the basal layer before they lift off. And the question that I'm really interested in thinking about next is what's going on in the stroma and how this relates to this rapid healing that we see. And so I'll stop there, and I mentioned all the folks who did the work as I was going through. I really want to acknowledge our collaborators, Rich and Adam, as well as Fred with the Worm Project, and Ophir Klein and Rappa, who is in his lab for the single-cell RNA-seq, and also our funders, particularly CIRM, NIH, and now we have funding from the tobacco program for the oral mucosal work. And thank you for your attention.
I have a question about the first part. So you mentioned that in a worm infection, the cells lose, so they're not anymore dependent on Wnt signaling, right? Or they're not sensitive to RSPO withdrawal, right?
So no, sorry. What I meant there was that if you take spheroids and you put them in high, if you take organoids, you put them in high Wnt media, then you'll see that they convert into spheroids. But we don't see in the intestinal, in the infection-derived spheroids that there are upregulations of Wnt targets.
So I was because I was curious if the mice would spontaneously develop cancer, because this is very so similar to cancer.
Yeah, so that's an experiment that somebody in the lab is doing right now, is to take mice which have a propensity to develop tumors and see if we infect them, what happens.
Like which model?
Like, for example, the APC min mouse. So yeah, we don't know yet what it's going to show, but we're interested in looking at that.
Thanks.
Hi. I'm interested in the epithelial turnover in humans versus mouse. There's a lot of discussion about the brain and the fact there might not be as much turnover in the human. What's the story with human epithelium in the gut?
So my understanding of this is that the turnover time is relatively similar, actually, in the order of days. That's a good question. You read that in the literature. I don't actually know all the details of the experiments that have been done, but that's the sort of consensus in the field. We work on this exclusively in mouse right now, so yeah, that's a good question.
Ophir.
Yeah.
I might have missed this, but the fetal versus the adult crypt program. So if we were able to label the crypt above the worm versus next door, how do they shed at the same speed?
So we have been really interested in following both the behavior of those cells as well as trying to be able to isolate them later and see what happens to them after in later periods of the infection or after the infection resolves. So we actually got a SCA1 Cree mouse, and unfortunately we were having some technical problems with it, so now we're trying to remake that mouse, but that's the only way that we can think of doing that experiment right now. So yeah, something we want to figure out.
And why would a worm change that program? What's it for them?
Oh, so like what the explanation for this might be?
Yeah.
Our speculation is, especially because we see these kinds of responses with other injury models, that something in the fetal gene expression program enables either increased healing through increased proliferation, or perhaps making more progenitors that you would use for healing, and that's why we think it's a kind of general response. There's a number of different approaches we could use to try to test that. For example, turning off the genes that we see in the fetus and see how that affects healing. So we're starting to do some of those experiments now.
We have one last question, though, so we have some time for lunch.
I was wondering, in the intestine story, is there, you were mentioning like a suppression of the CBC program. Is it that CBCs are lost, or is it truly a transcriptional dedifferentiation? And then kind of the second part of that is this just a expanded dedifferentiation response similar to reserve stem cells, but it's just kind of happening within the entire crypt, or is this truly unique compared to that?
Yeah, so those are both really good questions, and kind of like the ones that Shin was asking, they're a little technically challenging to test. So for the first one, Icebrand did do lineage tracing of LGR5 expressing cells and to see what happens to them. The problem with that experiment is that, and there's probably some clever way we can think of doing this, but we haven't figured out yet, is that to get the timing of induction of Cree activity versus when you're assaying is really hard. And your second question was about dedifferentiation, and the short answer is we don't know yet. What we don't see is cells. We don't see any obvious cells kind of falling back in, like you might in other scenarios, to turn on. We haven't formally proven that yet. I think one piece of evidence that speaks to this is that because it's such a general response, and including in some very drastic injuries where you're eliminating a lot of different cell types, that I don't think that this is the same kind of dedifferentiation that we see, for example, when we ablate the LGR5 expressing cells. But actually, that's a project that somebody in the lab has right now, is to try to compare across all these injuries. One thing that we've been really surprised by is just every time we have one of these unexpected responses is the breadth of behaviors that this tissue can exhibit. So I'm a little reluctant to speculate onto exactly what's going on, but I think it's a really interesting question.
All right, let's thank everyone.